My name is Dionne. I'm in the Material Science and Engineering Department, and I'm here with some of my coworkers, including postdoc Amr Saleh and student Xingxing Ho in EE and Applied Physics, respectively, to tell you about some of our work that we've performed in the first year of Catalyst funding on detecting bacterial bloodstream infections. While we're presenting the work, I do want to mention that this is a collaboration involving people both working on the hardware and the software and the business aspects. Many of the people are sitting over here, and they'll be able to answer questions after the talk. Okay, there's an old saying that says, all press is good press. And when it comes to global pandemics, I feel like viruses like Ebola and influenza and HIV pretty much get all of the credit. Meanwhile, bacteria pretty much remain the underdogs of the media, and yet they have a global impact. So in the US alone, if we're thinking about bacterial bloodstream infections, they cause more deaths than breast cancer, colon cancer, prostate cancer, and HIV combined. And then when we look to global health, in developing countries, about 30% of all deaths are due to bacterial infection. And such dire statistics are due in large part to the way bacterial infections are diagnosed today. So if we look to the typical procedure that's followed anywhere from developing countries all the way to here at Stanford Hospital, if you were to present with something that appeared to be a bacterial infection, especially a bacterial bloodstream infection, what would happen is they would take a sample. So in the case of a bloodstream infection, they would draw roughly about 5 milliliters up to about 20 milliliters of blood. And then what they would have to do is culture the blood to see if bacteria are present. And that can take anywhere from 1 to 40 days, depending upon the type of bacteria. So usually slow-growing bacteria, like tuberculosis, take much longer to culture. And then based on whether bacteria are there or not, there's usually a pH sensor that changes color, and that would detect whether a bacteria is there. Once they know if a bacterium is present, then they have to screen for antibiotic susceptibility by basically placing antibiotic patches over the bacterial cells to see if the cells survive or die. So meanwhile, you're in the hospital, not feeling quite well, and a doctor wants to be able to act on your infection. So according to the CDC, about 50% of patients will be treated with antibiotics and are unnecessarily treated with them or treated with the wrong type or dose. And that's something that's giving rise to emerging antimicrobial resistance. So what our team is trying to do is basically enable a so-called microbial culture shift, where we move away from this sort of culturing standard and use a new approach to rapidly identify bacterial infections in blood. And the idea is basically to take the few bacteria that would be present in a bloodstream infection, usually about 1 bacteria per mil, and be able to detect the very weak optical scattering that might come from that bacteria. So the idea is you would take the blood sample, basically print it into a 2-dimensional array where each pixel of that 2D array corresponds to a single cell. So that could be a red blood cell, a white blood cell, it could be a bacterial cell. And then we use optical scattering to detect which type of cell is in which pixel. So this is a process called Raman scattering. And you can imagine, since we're detecting scattering from a single cell, that the signal is going to be pretty noisy. So we need to combine machine learning with some hardware to amplify this signal. I think one of the advantages of this approach, especially compared to what's out there in industry, is that it requires no sample-specific preparation or cell tagging. So it's agnostic to the type of bacterial infection that a patient might have. So there are 5 objectives that our team has been working on in the past year and plans to move forward in the next 2 years. The first of which is designing these single-cellular microdroplets that efficiently scatter light, so that way we can get a stronger color to our hardware. Secondly, we need to make sure we can design fast and efficient bioprinters that can eject the microdroplets from blood. Thirdly, we have to engineer wide-field Raman cameras. Raman is normally a scanning technique, but we want to make sure we can do this basically in a single snapshot or as few a snapshot as possible. Then we need to make sure that we can accurately identify pathogens and their antibiotic susceptibility, so that way we can identify bacteria and their response to drugs basically in one step, and that's something that involves new machine learning algorithms. And then finally, we want to make sure that at the end of our 3 years, we can actually build a prototype, and we want to make sure that it's something that has market potential. So we've been pairing up with members of the School of Business to make sure that we have a market entry point. So with that, I want to hand over the microphone to Xingxing Ho, who's been one of the students pioneering a lot of the work going into identifying Raman signals from bacterial species and strains in collaboration with Stefano, Ermon, and Neil Jean.
Thanks, Jen. So when we started the project, we wanted to make sure that we could tell apart different bacterial species and strains based on just their Raman spectra. So to do this, we developed a Raman database composed of many different types of bacteria. For each of these bacteria, we spread them across a gold-coated microscope slide and took measurements in a confocal Raman microscope. So that means that each spectrum comes from roughly just one bacteria that's in the focal spot of the laser. We did this for 31 different strains from 22 species, and these are representative of 95% of patient cultures at Stanford Hospital. So one of the issues with Raman spectroscopy is it takes a long time to acquire high-quality, low-noise spectra. So you can see for the spectra shown here, many of the features are very similar, even when you don't have a lot of noise. There are a couple that are pretty different from the others, but for the most part, the peaks are similar between bacteria of the same species and also bacteria of the same, of different species as well. So, but in order to get a very large data set, we need to use much shorter acquisition times, closer to 1 second. And these spectra become very noisy and basically impossible to tell apart by eye. So the classical way of differentiating Raman spectra is using some sort of clustering algorithm, such as principal component analysis. And so you can see for when you try to tell apart blood and bacteria, when you plot them on 2 dimensions and the separation between points represents how different the spectra are, it's very easy to tell these 2 groups apart. But when you try to tell apart bacteria of different types, all the clusters overlap with each other, and it becomes impossible to differentiate bacteria of different species and strains. So to approach this complex problem, we worked with our teammates in computer science, Stefano and Neil, who designed a neural network that inputs a Raman spectrum, filters it through 25 convolutional layers that serve to filter out the noise, and remap the spectra to a space where it's easy to separate different strains. Now, the output of this network is a classification of 1 out of the 31 strains that is the bacteria that the Raman spectra corresponds to. So to give you an idea of what's going on a more detailed level, we can look at just the first layer. This is the layer that looks directly at the Raman spectrum, and it takes a series of filters that scan across the spectrum that look for features such as an upward slope, a downward slope, or a peak. You can do this for many different types of filters that look for different types of features. Now, each layer above this looks at the layer directly below it and takes more of these filters, building up complex combinations of these filters to generate a representation of the spectrum. So in the classification layer, the output is a series of predictive scores where each number indicates how likely the network thinks that this spectrum corresponds to that class of bacteria. Now in the training process, we compare this to what we know to be the true class of the bacteria, which is represented just as 100% in the correct class. And we repeat this process for about 50,000 spectra that we show the neural network so that it can adjust its weights and filters accordingly to minimize the difference between its prediction and what we know to be the truth. So after we've gone through this training process, we then test the network on a separate data set of about 12,000 spectra. And the results of that test can be seen in this thing called a confusion matrix, where each element is a percentage of the number of spectra in each true class that get predicted by the network as either the same class, so an accurate classification, or a different class, so a misclassification. So you can see here, for the first 3 bacteria, these are different strains of the same species. So we can really achieve high accuracies at the strain level. So when we zoom this out to the full 31 set of bacteria, our average strain level accuracy is 95.4%, which is quite high considering how noisy our data is. But you also notice that I've grouped the bacteria into these colored groups and also by the colored boxes in the matrix. These indicate different types of bacteria where the doctor would prescribe the same antibiotic regardless of which bacteria the patient has within the grouping. So that means that all of the misclassifications that are contained within the boxes actually don't affect what treatment the patient gets. So we can really condense this down into a treatment accuracy confusion matrix, where our average accuracy is much higher at 98.3%. And actually, most of the errors that are present in this matrix arise from the fact that we've included a pair of genetically engineered bacteria. So this is a pair of bacteria where they're basically the same, except for one gene that codes for antibiotic resistance. And this is something that tells us that we're really sensitive to the resistance level, not just larger differences in strains. So this means that we really have the potential to predict what treatment a patient has based off of detecting just their Raman spectra. But we wanted to see how this translates to clinical samples. So with the help of our teammates in the School of Medicine and the VA hospital, Mark and Niaz, we gathered 30 patient isolates from 6 different bacterial classes. We took the pre-trained neural network that had already seen all of our previous data set, and we fine-tuned it on a small amount of clinical spectra. So once we did this, we got a very low rate of failed predictions. Only 1 patient out of the 30 got predicted a treatment that would have resulted in a failed treatment. All of the rest of the patients have either correct predictions or, in a couple cases, predictions of an antibiotic that would have been too strong for them. But the patient would still survive. So we wanted to make sure that we can do this for very few spectra, because in an uncultured patient sample, there are only a few bacteria. So we looked at the dependence of how the diagnosis accuracy changes as a function of how many spectra we use. Even with just 1 spectrum, we can get 80% of these patients with the correct diagnosis. And when we increase that number to 10 spectra, we get about 90% of the patients correctly diagnosed. So with this, we've really shown that we can diagnose bacterial infections for using just Raman spectra collected in 1 second from uncultured patient samples. And with that, I'll hand the floor over to Amr, who will tell you more about our ongoing work and upcoming plans.
Thank you. So now that we have seen how we can identify single cells, let me walk you through our diagnostic tools. So we have a disposable sample well plate that is coupled to acoustic ejectors. We fill these well plate with a mixture of our blood sample that we want to analyze and also plasmonic nanoparticles that are necessary to enhance the Raman signature even further than what Xinqing had shown you already. With the acoustic waves, we can split this bulk of blood volume into actually micro droplets that we can use to create a printout of cells, as you have seen. Once we have this printout, we can simply use a Raman camera or like a spectral camera that can take pictures of parts of this printout, and basically all the spectral information coming from each cell will be encoded in the pixels of this, in groups of pixels of this sensor. Once we have the spectra, we can basically get a report that tells us what infection is in the sample, what antibiotic to use, and what the exact strain in the sample. Over the past year, we have realized our first prototype for our acoustic printer. So as you can see here in the movie, we use acoustic waves to eject microdroplets from an open surface. This work is being led by Fariha, Kamyar, and Pierre. And as you can see in this movie, the main advantage of using acoustic ejection versus other bioprinting techniques is that we eject from open surface. We don't need a nozzle to control the size of the droplet or anything like that. So we avoid all the contamination and the clogging issues that are typical for the bioprinting procedures that people have been struggling with. We were able to print from various viscosities, ranging from 1 centipoise to 3 centipoise of fluids. And these are the typical values for plasma and blood. So once we can print from a straight liquid, we started to test our platform with actually cellular solutions. What you see here is printing of E. coli on an agar plate. These colonies that are forming on the agar plate are coming from individual microdroplets that we have printed with our platform on those specific locations. So that means that we actually, as we are doing those viability studies on the cells, acoustic ejection is very gentle on the cells. So we maintain the cell as we print them, and they can still grow. So that means that we can analyze the blood and any pathogen in the cell, in the sample. So our next step in this platform is to start to try whole blood sample and whole blood sample spiked with pathogens so that we are ready for the scanning. But as we are putting these pieces together to build our prototype throughout the next coming 2 years, we have been working with our collaborator in the business school, Supriya, to explore the market out there so that we can actually build a prototype that is relevant to the actual end users in the hospital. So we have been interviewing clinicians and nurses in the Stanford Hospital, the Indiana University Hospital, and the VA hospital to tell us what they actually think the problem they are facing could be and what they want a new technology to satisfy. And we have been learning a lot about how the samples protocols can differ between hospitals and how the sample quality could differ between a sample coming from the ER versus a sample coming from the ICU, and also how hospitals make decisions on when to adopt a new technology. So to sum up what we have shared with you today, we have showed you how we combine machine learning with Raman spectroscopy so that we can identify with very high accuracy any bacteria at the strain level. And by combining these two platforms together, we will able to achieve prediction of the antibiotic and antibiotic resistance with accuracy that is more than 98%, even without the running antibiotic susceptibility tests. And we have shared with you our first prototype for our Raman, for our bioprinter for living cells. And with that, I would like to thank the Stanford Catalyst for supporting our work, and also the Gates Foundation, who have provided us with some seed funding over the last year. And I would like to invite our team members here, and thank you very much.
Thank you for a great presentation.
I have a quick question. I work in the NICU with preterm infants, and I just wondered whether your machine learning results could actually help inform treatment rather than assume, since treatment with antibiotics is still an art and a science, assume that they were getting the best treatment. Could your results actually help to inform more refined treatment?
Yeah, that's a good question. So when we're designing the network to predict treatments, we do take into account what the first choice antibiotics are. So we worked with collaborators at the Stanford Hospital to pick out for each infection what would be the best antibiotic if the doctors knew with 100% certainty what the bacterial infection is. So we do try to avoid cases where we prescribe broad spectrum antibiotics that aren't so good for the patient.
I'll also just add to that with our microdroplet platform, we have the ability to add in different antibiotic additives and see exactly how single cells are responding to those treatments. So we have a new student who just joined our team, who I think is here, Loza. Can you wave hi? Yep. So she'll be working with Sindy Tang to use our microfluidic platform, introduce antibiotics, and see how at the cellular level the cells are responding.
At the beginning, you talked about the fact that in developing countries, I think it was 50% of deaths were due to bacteria? 30%, a very large number. I thought I remembered that one of the goals of the project is to figure out how to deploy this technology in developing countries. Could you comment on that?
Okay. Does anyone want to answer this?
We were just going to mention that Manu Prakash is part of this project, and I think once we get a little bit further in the technology, then that will come into the aspect. So he's not been involved in the first year, but will be in second and third years.
So how much more accurate does your spectral analysis need to be for this to be commercially viable?
Yeah, that's something that we're also sort of trying to understand in sort of the business aspect of it is what accuracy do we need to get to in order to actually change doctor behavior? And it's a trade-off between accuracy and the speed at which you get the information. So if we are able to get, 98, 99% accuracy within the first day, it could change doctors' behaviors. Because the current state of the art is extremely accurate, but it takes several days to get. So, there's a certain benefit to getting the information earlier.
There's also different levels of it. When a person comes in septic and it basically could die in the next 6 to 8 hours, that's a little different than when someone has an infection that can be treated carefully. And so I think what's going to have to go forward are prospective clinical trials. But there's also another aspect, is that you can also modify this. For example, if there's a change in not picking up the methicillin-resistant Staph aureus, you can actually use a nanoparticle that is specific for that to increase the sensitivity. So even though we're using non-specific nanoparticles right now and label-free, you can add some labels in later when you find out where your deficits are.
Any other questions? If not, let's thank the team for the amazing work they've done.